Okay, so Biology 107 today. We're going to finish up topic 4. I have a code for you. And then we're going to start topic 5. And that should cover all of today. So let's talk about membranes, and then we're going to talk about cell walls. So cell walls are a little bit more interesting than membranes, I think, anyway. So let's do this. So last day, we had finished talking about some of the different types of transport, passive transport, active transport, and we just started getting into talking about bulk transport. So exocytosis and endocytosis. So some of these slides I'm just going through quickly because we already covered them. So exocytosis is bringing something out, endocytosis is bringing something in, and they're both doing this by basically manipulating their membranes and making these little vesicles that either fuse with the membrane to expel something or form as they're bringing something in for endocytosis. So we talked about phagocytosis, which is cellular eating. So they're bringing in something to eat, or this also could be an immune cell that is destroying something. So you can see there is our cell, and it's making these pseudopodia. So I talked about these. One is a pseudopodium, and many is a pseudopodia. And you can see it's making a food vacuole there in order to digest whatever it is that it's taking in. And there's an electron micrograph, and I think I showed you this video, and I think that was probably the last thing we saw last day. So we're going to move on from this, and we're going to talk about pinocytosis now. So phagocytosis, hold on a second, I feel like I missed a slide here. So just give me a second here to see if I can find it. Yes, there it is. Okay, sorry, I will just find that for you. I guess I'll just have to reshare this. Just a moment. What I wanted to share with you was a cartoon. Where is Zoom? There's Zoom. Okay, share again, and here we go. So I'm going to share a cartoon with you. Calvin and Hobbes, great cartoon if you're not familiar with it. It's a great cartoon. I spent a lot of time with Calvin and Hobbes when I was studying. Good relief. And check this out. We've got Calvin is under a blanket. He's pretending to be a giant amoeba. And he's crawling across the floor, and he's extending his pseudopod. And he's going to go for those cookies. So now you can appreciate this cartoon a little bit more because he's changing the shape of his membrane, or at least he's pretending to anyway. He has a good imagination. Of course, Mom is like, yeah, nice try, buddy. Put him back. So I thought you might enjoy that one. So pinocytosis is cellular drinking. So in this case, it's not usually necessarily bringing in water, although sometimes, often we're talking about bringing in solutes, also non-specific. So I have a video of this. There's an electron micrograph from the textbook. And you can see the same thing. You're just forming these membrane pockets and bringing in solutes and sometimes water. So this was kind of the best video I could find. It's not amazing. I'll play a little bit for you. It's 11 minutes long. We're not going to play the whole thing. Let's see, I think I got to skip up ahead a little bit. Here we go. And I'm not even sure what kind of cell that is, but you can see it's kind of blobbing around and bringing in something. Like I said, I don't know a lot about this particular video, but it kind of shows the process, and you can imagine it's forming those pockets and bringing in the solutes of some sort. So there's one other type of endocytosis that I wanted to talk about, which is called receptor-mediated endocytosis. It's a little bit of a mouthful, but it does mean something. This whole receptor part really means that it is bringing in a very specific molecule. If you take a look at this little diagram here, we've got these receptors here in red, and they're binding these particular purple things, whatever that is. Purple ligand basically means a purple molecule. And so you can kind of think of this as picky eating. So it's actually a different process in terms of how these pockets are formed. You can see they have these coated pits and all these kind of things. And I'm not going to get into all the specifics on that. You can read about it in the textbook if you like. But a good example of this is one way that we eat our body absorbs cholesterols. So you may have heard of HDL and LDL cholesterols, and you know, one of them is good and one of them is bad. It doesn't really matter, but this is how our cells actually absorb them, is they have specific receptors that will take in those lipids, and without those receptors, well, some people actually don't have the receptors, and it's a pretty serious genetic disease. Okay, so a little cartoon. This was the only video I could find on this. It's not even a video, but you get the idea. You've got a receptor. It's eating something in a picky way because it's taking a very specific type of molecule. So there they are: phagocytosis, pinocytosis, receptor-mediated endocytosis. So make sure you know what these things are. There'll definitely be a question or two on the exam. I'm probably not going to do a long answer question on this kind of thing, but just some words that you should know. Definitely phagocytosis is a word that comes up a lot in biology when you're talking about the immune system, and a number of other biological processes are rely on phagocytosis. And we're going to come back to phagocytosis as well when we talk about organelles and lysosomes and how we digest those things. So more on that coming soon. All right, so quick quiz question for you. It says, which statement is false, or which statements are false? That means we've got to go through them all one by one. So exocytosis and endocytosis are forms of active transport. That is technically true. If you think about the definition of active transport, it means it requires energy, right? And these actually require quite a bit of energy because they are transporting very large things. B, facilitated transport requires energy. No, it does not. Facilitated transport is a type of diffusion. C, facilitated transport requires channel or carrier proteins. Yes, that is true. Facilitated means helping, and what is helping are these little proteins. And then the last one, osmosis is a form of passive transport. That is true. Osmosis is specifically a type of transport involving water, and we like water. It helps keep us alive. Okay, so what I have for you now is I have a little video. This series called BioFlix, they come with the textbook, and the files are much too big to put on Moodle, but what I have is sometimes they show up on YouTube, so I have them linked in the lecture PowerPoints, and you can watch them that way. They're all about 3 minutes long. They're using the same graphics that are used in the textbook, and they're actually pretty high quality in terms of the animations and accuracy and those kind of things. So I have a bunch of these over the semester I'll share with you. Here's the first one, and it's kind of a recap of membrane transport. So I'll play that for you right now, and I think I have the sound enabled on Zoom, so you should be able to hear the video. If you can't, please do let me know. I'll press play.
We get all our energy and organic...
Oh, sorry, I accidentally touched my mouse. Try again.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, hopefully you enjoyed the cute little video. Sometimes maybe if you watch them carefully, there's some little jokes in there, but you gotta watch carefully. And you're gonna see some of those processes we're gonna come back and talk to again and again. So for example, protein export and you know, what's going on there with things getting secreted. So more on that later. So a few more things about membranes to consider. And then we're going to talk about cell walls. First is that this model here, this fluid mosaic model proposed back in 1972 is really the current model of the membrane. There are lots of kind of little updated versions, but they're just modifications on this previous one. You can see here's an updated version that's been widely published by somebody named Engelman. And he's talking about how membranes are directional, that there's a definite inside and outside, but it's still the fluid mosaic model. So if you do more studies on membranes, you'll learn about some of these things. Something else that we're going to talk about, not next unit, but the following unit, we're going to talk about eukaryotes. So that's topic 6. And of course, membranes are a huge part of many of these organelles. And the endomembrane system in particular is a huge, massive system, including the nucleus, the plasma membrane, the Golgi, the endoplasmic reticulum, all of those things are, of course, main part of them is membranes. So more on those later. And membranes move stuff around. So you can see in that video, you saw these little vesicles or vesicles. Doesn't matter how you say it, but these are little pockets that carry things around. So sometimes they're pockets formed you know, by the endoplasmic reticulum. Sometimes they're formed by the Golgi body, and they shuttle materials around the cell. So again, like I said, just that whole theme that cells are dynamic. They're moving and they're always doing stuff. And they're not just sitting still like this diagram looks like they're sitting still. Also, membranes are an important part of some organelles for things like certain enzymatic reactions. So the mitochondria is a good example of that, where it has all sorts of internal membranes. These internal membranes, they kind of look like little fingers here. I'm looking at these things here. And those actually have a name. They're called cristae, or the inner membrane. And that's where we have our electron transport chain. So huge, huge important structures for energy production in the cell. So here is our updated model of the membrane. And it's got those proteins, the peripheral proteins and the integral proteins. You can see the carbohydrates are in there. And we have lots of other stuff going on. And so, for example, on the inside, we have the cytoskeleton. And we're going to talk about the cytoskeleton, and that's going to be topic 7. So that's actually going to be after the midterm. So don't worry about that one at the moment. But what we do want to do is talk a little bit about what's going on the outside of the membrane now. And this is the cell wall and the extracellular matrix. So let's talk about what these things are, okay? I think I have some definitions on the next slide, we'll take a look at those. But we've already talked about the membrane. Remember, the membrane is this phospholipid bilayer. It's got proteins. It's involved in transport and a bunch of other things. So the other two things, the cell wall and the extracellular matrix, they're over here. They're outside of the membrane. And so they're adding to it. They have different functions, and most of it is structural kind of support functions. So let's see what I have for these definitions. So the cell membrane, all cells, the cell wall is rigid. So if you think of the word wall, a wall in your house, it's a pretty strong structure, and that's the same way as a cell wall in a cell. Extracellular matrix is something that's kind of a little bit more unique, and we're talking about just animal cells and just some animal cells. So we'll come to that in a few minutes. So let's talk about cell walls. Like I said, I like cell walls a lot more than cell membranes, but maybe that's because I did my PhD on the bacterial cell wall. But they're very important structures. So the one that you're probably familiar with is the plant cell wall. So if you zoom in on this picture, I'm zooming in right there. We're actually looking at 3 cells in this picture, right? So the main one is right here. This is number 1, and there's one over here, and there's one over here. And so you can see those 3 green things. We've got 3 cell walls. Now the cell wall is not green, by the way. You probably know what is green in a plant is the chloroplasts. Cell walls are actually relatively translucent. They're made out of cellulose. But we like to color them green because plants are green, and green is a nice color and all that, and it just shows up better rather than just making the whole thing white, right? But if you think of something like cotton or paper, these are things that are basically 100% cellulose, and they're white. So like I said, you're looking at 3 cells there, just to show you what's going on in the picture. And the cell wall is kind of a box shape, and so therefore the cell is a box shape. So whatever shape the cell wall is, that's the shape of your cell. And so it's forming structure, it's protecting it against osmotic pressure. So remember we were talking about last day about cells filling with water and some of them bursting. Well, plant cells don't do that. They like water. So let's take a closer look at what is going on in the plant cell wall. So first, it's made of cellulose, and we talked about cellulose already. I think I showed you this picture, and I definitely showed you this picture. And what I told you about cellulose is that cellulose is made out of glucose. So we could call cellulose a glucose polymer. And cellulose has a special type of linkage called a beta-glycosidic linkage that forms all of these hydrogen bonds. So you've got hydrogen bonds here and here. All of these dotted lines are hydrogen bonds. And hydrogen bonds are about maybe the quarter of a strength of an ionic bond. So they're weak, but when you have thousands of them, it adds up and you have kind of a tough structure. And that's what cellulose is. I know I also showed you this one. This is the one from the textbook, and I just don't think it's quite as nice showing the hydrogen bonds, but it does show down here the little fibers, and these are insoluble fibers. And so, you know, that word fiber, right? You hear about people wanting to get fiber in their diet, right? And this is what we're talking about. We're actually talking about cellulose. We can't digest it. So fiber in your diet, you know, even if you can't digest it, it's actually good for your digestive system. What it does is absorbs moisture and, you know, not to be too graphic, but it makes your stool a little more loose and not rough. And so this is why old people like to eat lots of fiber, because they're concerned about their bowel movements. And it's a good thing to have fiber in your diet. It actually encourages good bacteria in your gut too. So let's take a closer look at what's going on with that cell wall. Here's an image. I'll show you the textbook image in a minute, but you can see all of these kind of bluish things here. So I'm looking at this thing and this thing. That's the cellulose, and it's a tough structure. And you also have other things in that cell wall. So we have things like pectin as well. So some of you may have heard of pectin. If you've ever made like jam or jelly, sometimes pectin is one of the ingredients that you add, and it's kind of like a solidifying agent, right? So if you make jam and jelly, you're basically extracting juice from some sort of fruit, and you add pectin. Sometimes people might add, what's the other thing? I can't think of, a gelatin, right? A gelatin is an animal product. Pectin is a plant product. And you boil it, and then it's you know, turned into kind of a jelly afterwards. And the pectin is doing that. And the pectin is a little bit more, you can think of it as more glue-like filler, right? So whereas the cellulose is really the strength of the entire structure. So it's kind of a sticky filler, I guess you could think of it as. So notice this is outside of the membrane. This is the membrane here, and the cell wall is outside of the membrane, protecting the membrane. So it's a protective barrier, right? So let's take a look at the textbook image, and I'm going to draw you a little bit image on the right here, and it's not going to be amazing, so I apologize to you for that, and I might have to do some erasing because I'm trying to show you how these things grow. So if you take a look, just like that other picture, we're zooming in on 3 cells, and you can see the third one is kind of barely down over here, and then we've got 1 here. So maybe I'll just number them. So we've got 1 and 2, and the third one is kind of like right there. So what are we looking at here? So first of all, we're looking at something called a primary cell wall. So actually, let me draw that for you. And so I'm going to draw just down here in the corner. So I'm going to draw like a membrane. So we'll call this M for the membrane. Okay. And what happens when a plant grows? And like I said, unfortunately, I'm going to have to do some erasing on this because I'm going to show you what happens when it grows. But what happens is it makes some cellulose. And here's my cellulose over here. And so that's my primary cell wall. Okay. And that's what happens at first. Okay. Now, what happens next? We'll come back to that middle lamella in a minute. Okay. But it's kind of just sticking cells together with lots of pectins. And then a secondary cell wall grows, right? So what happens is this primary cell wall, so I've got to erase now for you. Okay. So the primary cell wall kind of gets pushed away. So primary cell wall. And then the secondary cell wall fills in here. So I'll try to make this a little bit of a different image like that. So there it is. There's my secondary cell wall. So the secondary cell wall is actually closer to the membrane. And you can see that here, right? You know, if I'm looking at, let's look at cell number 2. We've got the cell membrane right here. This is the membrane. This is my secondary cell wall. And then I have my primary cell wall. So the primary comes first, but it gets pushed away from the cell membrane. So there's some, you know, that's just kind of the way it works. And the secondary cell wall is not necessarily found in all cells. A lot of plants have it, but not a lot of plants do not as well. And often it's called wood or structures kind of like that, where the cells need to be extra strong, usually for stems or branches and things like that. There's other words besides wood. I just can't think of them at the moment. What the names of the structures are, like xylans and other fibrous structures anyway. So this middle lamella, like I said, is just kind of contains a bunch of pectins and is kind of gluing the cells together. One other structure I want to point out, so another word for you, are these plasmodesmata. So these are basically little channels between the cells, and actually the cytoplasm is connected. So if you think about a tree, it's going to get its water from the roots, from the ground, and the water has to diffuse all the way from those roots all the way up and all the way to the leaves. It's a long way. So rather than having to go through membranes, they make little channels, and it speeds up the diffusion. And so this happens in a lot of plant tissues to kind of just allow the diffusion to be a lot more rapid. I think I got an electron micrograph of those. There we go. And you can see basically, like I said, it's the membranes are connected. They're little channels, and those cells are directly connected in between each other. There's also other things that go through there. Plants have hormones, by the way, and they can transmit cellular messages through those channels as well. So that is the plant cell wall. I want to talk about some other cell walls, and there's a whole bunch here, and I have some stars that are the ones you need to know. Okay, so plant cell wall for sure. You need to know a little bit about it. Know it's made of cellulose, and know some of those definitions I just gave you. We're going to talk about animals in a few minutes. They don't have a cell wall, but some of them do have an extracellular matrix, which is kind of an analogous structure. Bacteria, most of them have something called peptidoglycan. That one is important. Archaea, they don't have peptidoglycan. I'll talk about them in a minute. but they have something sometimes called pseudopeptidoglycan. Fungal cells have chitin, and protists, there's a large diversity. So what do you need to know? I'm not going to ask you about archaea cell walls on the test or protist cell walls, but I could be asking you about the bacterial cell wall or the plant cell wall or animal extracellular matrices or fungal cell walls. So some words you need to know, and we've seen these words already. Okay, we saw them in topic 2 when we were talking about macromolecules. So I told you we would come back to them. And so here we are. So let's talk about peptidoglycan. So peptidoglycan, as I mentioned way back in topic 2, is a carbohydrate, and it is a lot like cellulose, except for it's very decorated. So if you take a look at, for example, I'm looking at this one here. That's basically, that's glucose, except for this glucose has this group on it. This is an acetyl group, and this is a nitrogen. So we call this N for the nitrogen, acetyl for the acetyl group, and glucosamine because it's got glucose in it, and the amine is just the NH, right? So we call it NAG. I won't ask you about N-acetylglucosamine on the test, but you should know that peptidoglycan is a carbohydrate that has peptides on it, okay, because that's in the name. That other carbohydrate over here on the left, N-acetylmuramic acid, you can call NAM, and it's kind of unique in that it's got these little things here. So if you know what those are amino acids, and those amino acids form a little peptide. And this little peptide can link up with another peptide right here. That's called a cross bridge. And so what we end up is, rather than a bunch of kind of just linear fibers, peptidoglycan actually makes a three-dimensional matrix. So you can think of it as something that looks like that. And so these are actually massive molecules that surround the entire cell in whatever shape that cell is. So it could be a rod-shaped cell, it could be a round-shaped cell, and the peptidoglycan is one strong structural molecule around the bacterial cell. So it's protecting the bacterial cells against osmotic pressure, kind of like plants, and it's a lot skinnier than cellulose, so it needs to be a lot stronger as well. So there's lots that could be said about the bacterial cell wall, especially for me, because I know lots about them. But one thing that you should know is that there are two types of bacterial cell walls. And so if you take a look, we classify most bacteria into one of these two groups. We call them the gram-positives or the gram-negatives. So I mentioned when we were talking about microscopy that we'd talk about this a little bit, and we'll talk about a little bit in the lab as well. And so this Gram thing is named after a guy. His name was Hans Christian Gram. And he was staining cells and found out that some of them stain purple using this certain procedure. So he called them gram-positive. You can see I even have the font in purple. And some of them didn't take up the purple stain in the same way. So they were named gram-negative, and they actually stain kind of a pinkish reddish color. So you can see maybe in the little micrograph at the top, there's purple round cells and long and skinny reddish pinkish cells. So a couple of things to say about this. The structures are very different, and that's why they stain differently. So if you take a look at the left, the gram-positive, it's a lot like a plant cell wall. You've got the plasma membrane, and then you've got a bunch of peptidoglycan. So this is the peptidoglycan here, PG, right? So it's kind of a thick layer of that carbohydrate and peptide stuff, right? And it, like I said, looks a lot like a plant cell wall, other than the fact it's a different carbohydrate. The gram-negatives, on the other hand, are extra kind of unique. I kind of think of them as a sandwich membrane. So take a look, we actually have two membranes. We have a plasma membrane, and then we have something that we call the outer membrane, so OM. And I'll talk about the outer membrane in a minute. It actually has some unique lipids in it as well. And the peptidoglycan is kind of a thin little layer on the middle, and enough to protect the cell, and but a much thinner layer than what you'd see in the gram-positives. So here's some micrographs. These micrographs actually were taken by a friend of mine at the University of Guelph, and he actually was one of like 3 people that could do this special technique and got these amazing pictures. So I just had to share them with you. In fact, look at these pictures. I'm looking at the gram-positive on the left. You got the thick peptidoglycan right here, And you can clearly see the phospholipid bilayer there as well, which is really quite remarkable. On the right, you have the gram-negatives. You can see two membranes, so the outer membrane and the inner membrane, and that thin layer of peptidoglycan in the middle. So really, really incredible pictures. So let's talk a little bit about the staining technique, okay, and how it works. And we'll talk about a little bit more in the lab in lab 4. So that will be next week, I guess. So the gram-positive, I said it takes up the stain. It's got this thick layer of peptidoglycan, and the stain will go in there and okay, hopefully you can hear me now. I don't know what happened with Zoom. It just gave me this notice. Maybe I clicked something by accident. I'll have to make sure I reshare here. Okay, sorry about that. So the gram-negatives, the crystal violet stain, it just doesn't get in there because it's got that extra membrane that kind of protects it. So we throw in a second stain, and the second stain is called saffron and red, and so the gram-negatives end up that kind of pinkish reddish color. If you have a lot, it turns more red. If you have less, it kind of turns pinkish. I'll show you some images here. So here's a gram-positive on the left. You can see a nice dark purple color, and E. coli is gram-negative on the right. So we'll get a chance to look at lots of these photos for lab 4. So there's one of the photos I took. Sometimes it's hard to get really sharp pictures when you're zooming in on something so small, but you can definitely see the two different types of bacteria there. The gram-positives are purple and the gram-negatives are pink. So another couple of things to say about gram-negatives. There's a lot of really important gram-negative pathogens that make us sick. And so, you know, we're wondering what's going on here. And there's a couple of reasons why they might make us sick. One is this outer membrane here. This outer membrane has a unique lipid. It's called LPS, or lipopolysaccharide. And it's unique in that only gram-negative organisms have it. And the thing that is important about the lipopolysaccharide to know is that it can make us sick. So if you get it in your blood, let's say you get a bacterial infection in your blood, your immune system recognizes this as something that's a foreign molecule. And you can have a severe immune reaction in some cases. Most people, it's, you know, you get, your blood pressure lowers and you get a fever and a few things like that. But if you're already really sick, like if you're an old person or a baby, you know, these people are at high risk group of actually dying from this LPS being in their blood. So in some cases, it's well, in most cases, it's very dangerous to have a bacterial blood infection. The other thing that is unique is we've got this new zone. We've got this little tiny, skinny little layer between the two membranes. It's called a periplasm. And it turns out the periplasm, there's a few extra enzymes and things like that in there. And this is an area of concern of some bacteria because they have enzymes that can actually break down antibiotics. So it gives them resistance to certain drugs. So anyway, just something a little different with this gram-negative, very unique kind of membrane cell wall system. So what about the archaea? I mentioned I'm not going to test you on the archaea cell walls, but it's just kind of worth it to talk about just for a minute. They do not have peptidoglycan. Some of them have a carbohydrate that, if you're not a biochemist and you just look at it quickly, you might think is peptidoglycan. But so it's close enough that sometimes people call it pseudo-peptidoglycan, but it's not peptidoglycan because they're not bacteria. Some of them have these S layers. S, by the way, stands for surface. And these S layers, so S is for surface. I'm just going to write that, surface. And these are actually proteins that crystallize. And you can see there's an electron micrograph showing an archaea there. So they have some unique things going on because they're archaea and they're unique, and that's the way they like it. So what about fungal cell walls? I mentioned before fungal cell walls are made out of chitin. And chitin is a lot like cellulose, but it has N-acetylglucosamine instead of glucose residues. And other than that, it's very similar to a plant cell wall. It's got the chitin on the outside. It's rigid. There's some other compounds, carbohydrates and proteins in there that are kind of like the pectin. They're kind of like the glue. But other than that, very, very similar situation going on. So what about protists? I haven't talked a lot about protists yet. We're going to kind of talk about them here and there over the next few units. But remember that protists, there are eukaryotes, and most of them are unicellular. And other than that, there's just a huge amount of variety. Some of them have cellulose, some of them have chitin, some of them have all sorts of unique different types of carbohydrates. So kind of the big thing to think about protists is to know that they're just unique, and there's lots of weird things going on with them. Some of them have these calcium carbonate things, and that's what I'm showing in these images. These really interesting crystalline structural cell walls, which are really fascinating to look at under a microscope. Okay, so the last part of this unit is to talk about the extracellular matrix. And I kept telling you we'd get to this. So animal cells don't have a cell wall. We don't have cellulose, we don't have peptidoglycan, we don't have rigid things around our cells. But sometimes we have things that are semi-rigid, or, well, and there's a few examples where we actually have rigid things. But there's not really any one situation where we can say all cells have something, okay? Different types of animal cells may have different types of things outside of them. And when they do have things outside of them, we often call it the extracellular matrix. So in this diagram here, you can see kind of the most common type of extracellular matrix in animal cells. And this is comprised mostly of something called collagen. So collagen is, well, it's represented here by these long strands. These are rope-like proteins, and they're relatively flexible, and they're found pretty much everywhere in your body. So they're found all over in your skin, and that's what's giving your skin a little bit of bounce to it, right? So if I kind of, you know, if I poke myself with my finger, my skin is kind of bouncy, and that's what collagen does. It kind of gives a little bit of bounce to your skin. And so I don't know how old everybody is here, but if you are 20, this is one area that you are already aging. Somewhere around 18, 19, 20, our bodies actually start making less collagen. And as we get older and older and older, we make less and less and less collagen. And maybe you've seen, you know, elderly people, sometimes their skin is not bouncy, it's a little bit more fragile and all that. And that's because we make less collagen. You know, some people, believe it or not, I found this out, you can get collagen injections, and some people will do this to make their lips look fuller and whatnot, and I guess they want to look like celebrities and things like that. But that's what it's doing to your skin. So it's good stuff. But not all of our cells have collagen, right, around them. We've got all sorts of different types of cells. I'll give you one or two more examples of extracellular matrix. One example is bone cells. The bone cells, I'm going to draw a little cell. Here's a little bone cell. It's got a little nucleus. They're not even touching. They're actually pretty far apart. So there's 3 bone cells there. And what they have is all sorts of there's different types of calcium minerals. That's Ca2+. So let me just try to make that better. And they're all over the place, all around this. And it mineralizes and makes a hard structure. Something similar to bones is, of course, cartilage. And cartilage is a bit more rubbery, and they're not calcified. So cartilage is found like in your ear. Your ear is kind of firm but flexible, right? Same thing with the tip of your nose. It's firm and flexible. So I'm not sure what all the proteins are in there that are making that extracellular matrix, but it is kind of a firm structure. So somewhere between collagen and the calcified bone structures. Okay, I know I have this slide. I'm going to skip this. This is just talking about how cells are attached together. It's not really that interesting. Okay, so we are ready for a Kahoot. So let's load this up and we'll do the Kahoot. So load up your app and I will get it ready. Here we go. I just need to share it and we can play Kahoot. Okay, here we go. Hopefully you can see that now. And it should start loading the game number in a moment. All right, here we go. So game number 2506813. All right, I'll give you about 10 more seconds here. I think we got most People in there? Okay, away we go. You can join at any time. The numbers are going to be at the bottom of the screen. So 8 questions. Membranes and cell walls. Number 1, for the type of membrane protein that is labeled, what is it? Okay, that was a tough one. Started off with a tough one here. Most of the amino acids are hydrophobic. So take a look at the picture. Remember that this is an integral membrane protein, not a peripheral one. So this one here is wrong, peripheral. Because it's embedded into the hydrophobic layer of the phospholipids, most of the amino acids are going to be hydrophobic. So good job, whoever got that correct answer. Okay, so I guess that tells us who got that one correct. So good job. Number 2, true or false? Phagocytosis is a type of cellular drinking. By the way, the true and false, you only have 10 seconds, so answer quickly. And the answer is false. Phagocytosis is cellular eating, not drinking. Pinocytosis is cellular drinking. Okay, question 3, true or false? Facilitated transport requires energy. It should say requires, not required. And the answer is false. It's active transport requires energy. So make sure you know your definitions, or these are going to be tough. Okay, it looks like we've got at least one person on a hot streak. Number 4, cholesterol is what? So cholesterol regulates membrane fluidity. It is not a protein. Cholesterol is a lipid, and it is not part of the cell wall. It is part of the cell membrane. So make sure you know the difference between a cell wall and a cell membrane. Cell membrane is, of course, made of phospholipids and proteins, and the cell wall is what we just talked about. So we're talking about cellulose and peptidoglycan and chitin and a few other things. Okay, number 5, true or false? The fungal cell wall is made of cellulose. There's a diagram, and I conveniently cut out some of the labels. False, made out of chitin. So what is made of cellulose is the plant cell wall. Okay, a couple more questions, 6 of 8. A gram-positive cell wall has a thicker layer of peptidoglycan, spelling mistake, than a gram-negative cell wall. And the answer is true. So this is the gram-positive shown on the right, and remember it stains purple, and it looks a little bit like a plant cell wall, whereas the sandwich one is the gram-negative. Okay, question 7. Which of the following is true regarding the extracellular matrix? Okay, the green one is correct. So collagen is in connective tissue as an example. And number 4 is not true. Not all animal cells have an extracellular matrix. Your red blood cells don't have anything on the outside of them other than membranes. So there's lots of examples of animal cells that do not have an extracellular matrix. Okay, one more question, I believe. True or false? Plant cells like to be in a hypotonic solution. Oh, divided down the middle. So remember, hypotonic means there is less solute on the outside than on the inside. So it's true. Plants like to be in pure water, And so the answer is true. Okay, time for the final scores. With the bronze, T. With the silver, Mark. The gold, Levi. Well done. You started off with the lead and you kept it the whole time. So good job. Okay, so just give me a moment here. I'm going to switch things up, and I know there's not a lot of time left, but I think there's just enough time to talk about prokaryotes and eukaryotes. So let me just get to those slides here. Okay, Zoom, come on, you can share for me. There we go. Okay, so we've already seen this slide or a similar one. And so the next topic is prokaryotes. And so what I want to do now is kind of just talk about these in a little bit more detail. So what I have for you right now is I'm going to make a few notes regarding prokaryotic and eukaryotic cells. And then some of these things we've talked about a little bit, and some of these things we are going to talk about very soon. So hopefully this table will give you an idea of some of the things we're going to talk about and some of these differences. So hopefully remember that a eukaryote, U, remember, means true, and karyote means nucleus. Prokaryote means before nucleus. So if you're asked on a test what the main difference is, you better say something about the nucleus, because one of them has one and one of them does not. Okay? So I'm going to switch now, and like I said, this is usually something that I do on a whiteboard, but I'm going to do this actually on a Word document, so you can type or write, whatever works for you. And like I said, I don't post these, but if you really want to request it, I can always share this with you. Just send me an email. So here's my my table, and actually I'm going to go down to number 3, membrane-bound nucleus. So this is really a yes/no kind of thing, right? Eukaryotes have a membrane-bound nucleus, and prokaryotes do not. So what are we talking about for the prokaryotes? We're talking about bacteria and archaea. Okay, so we are going to talk more about what these archaea are next day when we have a little bit of time. The eukaryotes, we have animals and plants. Hopefully that's an easy one. We also have fungi, and we have protists. So sometimes people will call algae its own kind of group. Kind of when we talk about protists, it could mean a whole bunch of different things. And like I said, we'll talk about protists a bit more when we get to topic 6. So typical size of bacteria. So we're talking about pretty small, and this is, let's see here, I gotta get that special symbol. I'm looking for that one there, that's a micro. So 0.5 micrometers. Maybe I'll just put 0.5 to about maybe 5 micrometers is typical for a bacteria. So 5 micrometers, 1 micrometer is 1/1000th of a millimeter, so 1,000 times smaller than a millimeter. So these are very, very tiny. Eukaryotic cells, we're looking at the smallest ones are around 20, and we can get pretty big on some of these. In fact, some of them are much larger than this entirely. Like we have a neuron that goes from our hip joint to our big toe, and that's one cell. But that's kind of unusual. Most of them are kind of in the 50 to a couple hundred range. Some plant cells are quite a bit larger, so in terms of size. Other membrane-bound organelles, bacteria, no. Eukaryotes, yes. And this is one of these things I could put usually in front of this, because this is biology. You can put usually in front of every sentence in biology, and it's probably true. There are exceptions of some weird things that you can find in bacteria that you could call a membrane-bound organelle, but usually the case is no. Do prokaryotes have a cell wall? Yes, they do. Remember, bacteria have peptidoglycan. And archaea, need to spell. They could have pseudopeptidoglycan, or they can have S layers. Eukaryotes, so animals. No cell wall. Plants, they have cellulose. Fungi have chitin. So this is all stuff that we've talked about already. So I know that we're almost out of time here. I'm just going to fill in some details for these other parts here. And so sorry for going a minute or two over, but I feel it's important to fill in this table here. So bacteria have DNA. Yes, of course they do. Prokaryotes have DNA. But we're talking about a circular chromosome. Circular chromosome. And usually only one. Eukaryotes, we have many linear chromosomes. And you can talk about haploid and all those kind of things. We're not going to bother at the moment. And DNA is packaged by histones. So we're going to talk about histones. I don't think we're even going to get there until about topic 13 or 14. So don't worry about those at the moment. Ribosomes in prokaryotes are smaller, and in eukaryotes they're larger. So we'll talk about those. You probably know what reproduction looks like in eukaryotes. We have mitosis and meiosis. Whereas in bacteria and archaea, we have something called binary fission. Okay, and lastly, motility. Some have, some bacteria and archaea have flagella. And in eukaryotes, we have flagella, something called cilia. Some amoebas have amoeboid movement, and a few other things. So there's a bit more variety in terms of how eukaryotic cells get around. So I know that's a lot kind of to throw in there at last minute. Your brains are probably tired, but hopefully this gives you a good place to start so that next day when we come back and talk about prokaryotes and eukaryotes, you're going to see all these things are going to pop up. And this is actually one of the themes of Biology 107, is the differences between the different types of cells and why they're unique and special and all that. So a good place to start is what are prokaryotes and what are eukaryotes. One of them has no nucleus, the other has a nucleus. Okay, so 2 minutes overtime. Sorry about that. I'll do better next time. Happy Monday. We'll see you on Wednesday.